hemoglobin, it's polypeptides, right, that make the globin. If you don't have a nucleus in a cell with DNA, you can't make that stuff because the DNA acts as the code to, genetic code to make it. It's an involved process, of course, but you have to have a nucleus to act as the blueprint to make the protein. If you don't have one, you don't. Is that?
Yes, that makes sense.
Going to clear. Yeah, it did. Doggone it. It seems to be delayed on this thing. Let's screw it up. Okay, now we're going to blood type. Let me preface our discussion of blood types before we get into the types and what produces them. I think some of you know this already. We are concerned about blood types, particularly because of the T word. What is it? Transfusions, yeah. We can also be concerned about it concerning reactions between mother and baby that she's carrying too. We'll talk about some of that toward the end. But it's particularly with transfusions because we have different blood types. I'm sure you've heard of some of the types, and maybe you even know what your own is. But there are a whole bunch of different blood types. The main ones we're concerned about, as you can see up here, would be the ABO and RH. And if you're ABO type, if you have your ABO type, you'll find yourself either A, B, what are the other two? AB,
or O,
which is not very common, and O, which is most populations, most common blood type. And we'll see later that you can put with that the RH type. And if you look at your RH type, it's either one or two things. What is it? One or two.
positive and negative.
Or negative. Later on we'll put those together. We'll initially start out talking about them separately because when you initially talk about blood types, if you put both of them together, it can be extremely, well, it can be more confusing than it is anyway. Let's put it that way. So, the idea is you have people giving blood, and we'll talk about names of those, what we call those folks. You give blood to a person, and if that person gets the wrong blood, what you're doing is you're introducing into the receiver's blood, the recipient as they call them, we'll write that down later, you're introducing blood that is incompatible with theirs. And what will happen is their immune system, antibodies basically, is of the recipient now, will attack and clump those foreign RBCs and cause a tremendous reaction in the recipient's blood. So you make sure you transfuse the right blood into the person or there can be a problem. And as far as the compatibilities are concerned, we'll learn those. There's a whole table that I want you to fill out and we'll talk about it. You've got to understand the science behind blood types before you can understand what the proper transfusions are too, so we'll go through that, discussion of that. But let's first start talking about ABO type. This is where I've done some modifications here. We go back to our drawer again, hope this works okay. On the surface of red blood cells, there are a number of different chemicals. And they're what we call recognition proteins, actually. The chemicals can be called antigens, or agglutinogens. That's a G-E-N-S on the agglutinogens. See why they're called that in just a minute. But these are called antigens because if they are introduced into another person's blood and they're not the right ones, there are antibodies in that person's blood that can attack them. They become foreign invaders. Any chemical or organism that is non-self and is going to cause production of antibodies in a person, going to cause an immune response in the person getting those antigens, can have a problem, and they produce those antibodies to try to kill or destroy the antigen that's coming in. If you get the wrong blood in somebody, your immune system treats those blood cells as antigens, as foreign, and your immune system attempts to destroy them. But in doing so, if you get transfusion of the wrong blood, in doing so, and trying to destroy those antigens, your own immune system can cause you a tremendous problem too. So the proper name for them, the technical name would be agglutinogens. I generally refer to them as antigens because it's easier to say every time, but you must know that blood cell antigens are also called agglutinogens. The reason for that is the process of clumping the blood together in your system is called agglutination. And a way to remember agglutination is GLU is part of the word glue. And it kind of sticks, I know it's a long word, but it kind of sticks the cells together and destroys them. So that's what you call these guys. And I'm probably going to overlap here a little bit. Out. All I'm going to say about what gives you your blood type is I'm going to say your blood type is inherited. All right? The study of this inheritance is really interesting, but if we spent time on that in here, we would never get finished with this stuff in time. If you want to know more about the inheritance of blood type, I'm glad to sit down with you or in lab sometime and explain the whole deal. But it's fairly simple inheritance. You have to have some knowledge of basic genetics, all right? But just suffice it to say that what your blood type is, when you're looking at ABO or Rh blood type, you get it from your mom and dad. You determine what your type is. And you're not going to necessarily turn out to be the same type your mom and dad would be. Depends on what type you're talking about. Like if your mom is A and your dad is B, you could be any of the types of blood. Of ABO, that is. If both parents are negative, you're going to be negative. If one of them is negative and one of them is positive, you could be negative or positive. So it's the way the genes combine when the sperm meets the egg as to what your type will be. It very much is a genetic determination there. Wish I had more time to talk about it, but I really need to move on to the next topic. If you have any questions on blood types, please talk to me and I'll sit down and explain it to you. It won't take more than 5 hours. It's very, very easy. I think we've been through this before, we know it, but I still need to write it down here. If you're giving blood to someone, you've got no problem other than giving the blood. Be comfortable for you. But what do you call the person giving the blood?
Donor.
Very simple, a donor. And the person getting it, if I can squeeze this longer word in here.
Recipients.
The recipient. And when I say the immune system can or will or may have antibodies, that depends on the type of blood and the type you're talking about. For example, if your blood type is A, you cannot have antibodies against A. We will see that you will have another antibody. We'll talk about that when we get to the chart in just a minute. If your blood type is like Rh positive, you can't have an anti-Rh antibody, can you? And so, and the antibodies have a technical name too that I want to write down here. Try to squeeze it into this space with as big writer here. They are actually called agglutinins. That's T-I-N-I-N-S, agglutinin. Agglutinins and agglutinogens. Don't they sound alike? Agglutinogen would be the antigen, the stuff on the RBC surface. The antibodies or agglutinins are going to be in the person's plasma. They will not be on the RBCs. As antibodies quite often found, they're found in your, other places too in your body, but in your bloodstream, floating around within the plasma. So if blood cells containing a certain antigen are transfused into a person who has the antibodies floating in their plasma that match those antigens, say I'm an RBC and here's an antibody that matches it, it's going to bind chemically to it, bind to other cells around it, and those cells are just going to be all clumped and glued together, and there's a mess. And that's cross-reaction, as they call it. That's what you get when you get the wrong blood in here. And it's what blood typing is based on, too, where you put, like, I tried Tuesday and failed. Thursday worked perfectly because I had good, serums. But in typing my blood, I put 3 drops of my blood down, and I forced the issue where I put 3 different antibodies, the anti-A, anti-B, anti-RH in there. And the students on Thursday saw the clumping. Okay? But that's typing. Transfusion is different. That's where a person receives the blood they shouldn't receive. The resident antibodies in the person's blood will attack those cells, and you'll get the agglutination, you'll get the clumping, but it'll be inside the person's body. So remember, antigens on the red blood cells, antibodies in the plasma. That's where that's different. Got that point? What I want to do now is go through this. This is one of the things I switched around. You might see this table later. You'll look a little bit further in your list and you'll see the table. Because I want to explain the ABO type and then go on from there. Since I said these things are in the plasma, we'll see that on this table. I like organizing things into tables because I think it's a good way to summarize and learn. And so if you look at the ABO types, not the RH now, we're leaving that for later. We can put them together later. Right now we'll separate them. If you look at the ABO types, you know already you have A, B, AB, or O. And that'll be in this first column here, the type. If you look at the A type, you got a gene. If you're type A, you received a gene from one of your, maybe both, but at least one of your parents that caused you to produce this A antigen on your red blood cells. RBCs you're producing, you have a genetic code in there that says make this A antigen. And that's on the surface of the red blood cells. Type A, simple. A antigen is what you produce. What will you have in your plasma?
B and O antibodies.
Anti-B. Yep. Couldn't have anti-A because you would clump your own blood. But along the line, your immune system produced anti-B antibodies. And they would be in your plasma. And the same idea is for B. Same exact thing, but a different antigen. You've got the B blood type, you've got the B antigen. You know what's going to be in the next column. Anti-A antibodies will be in your plasma if you're that type. AB is simple. Remember, if you're B type, you get at least one B gene from a parent. 1 or 2 when you're B. Okay? If you're AB, one parent has to be A, the other has to have a B gene to give to you. Or I should say one parent has to have an A gene to give to you. It doesn't mean they're A type. But they would have to have an A gene to give to you and a B gene to give to you. Okay? And then you become AB because now you have both the A and the B antigen on your red blood cell. What antibodies can your blood have?
None.
ABO. Well, they can have others, but the ABO type antibodies, no. Neither one can be present, or you would clump your own blood. You can't do that. Then we come to the most common blood type of all. Now, if we knew everybody's type in here, that may not be true. It probably would be, but it may not. The O type is the most common. Okay? It may not be true. But if you get enough of the population, you will find that O type is typically the most prevalent in that population. And there are even situations where groups of people, the O type is almost totally predominant in the populations. Very common. But what you lack as an O type, you lack the ability, you genetically lack the ability to produce the A or the B antigen, so it would be absent on the RBCs. And therefore, what antibodies, ABO antibodies now, what ABO antibodies can your blood, your plasma contain? Both. That's the key to understanding the basics of ABO blood type. Knowing what antigen is there, and knowing that you have to either have the opposite or neither of the antibodies, depending. The reason AB can't have either one is because it has both antigens. If you had anti-A or anti-B, you'd be clumping your own blood. But the percentage goes for most populations, and I give you, your book has a nice table which shows different groups of people and what their blood types are, whether they be Oriental or whatever, you know, white, African types or whatever, what their types would be. Native Americans, that kind of thing, they differ. But basically, O, most common, followed by what? A, followed by B, and least common of all is AB. Typical percentage for AB might be like 4% or so of the population. Now RH is a little bit different. It's still a blood type. It's still produced by an antigen, by antigens that are present or not present, but the antibody thing's a little different there. Actually, you produce the antibody if you're exposed to it, but that's another story. I like this because this is a color picture showing the same thing that I just talked about. You can see up there the little purple lollipops represent the A antigen, and what you have to have is the NEB antibody. You notice the antibody has 5 prongs, or 5 points in 5 directions, so it can bind to a cell here. You can write those on there. It could bind to a cell like here. No, it can't. Sometimes I worry about my standing. It can bind to a cell here, here. And of course, there could be others over there, so it can just go on ad infinitum, bonding these cells together. But it would have to have, in this case, have to have the A antigen on the surface of the donated cells that enter this person's plasma. Okay? Now, if I gave A to B, you see this matches. If I give B to A, it matches. So you can't give these bloods. Can you give AB to anybody? No, because look what happens if you give AB to type A person. You've got the anti-A, anti-B antibodies, or you could have the anti-A antibodies. But for type O, look how clean it is. There's no ABO antigen, so hey, it could go to anybody. Be given to anybody. Of course, it can't receive